Okay, welcome to this lecture on the species concept. I'm Raphael, and this is part of the crash course in evolutionary biology. So in this video, we'll learn about this thorny issue that has been bugging biologists for centuries now, and that is, what is a species? Now, if you recall, in the video on speciation, we learned that a critical ingredient to the completion of speciation, and so the divergence of two species basically, is the establishment of some sort of reproductive isolation between them, some postzygotic barriers or some prezygotic barriers, something that prevents two populations from interbreeding. And that, we said, is essential to the formation of distinct species under the so-called biological species concept, a species definition proposed by Ernst Mayr, and which basically states that species are groups of organisms that are reproductively isolated from other such groups. Now, that is a very common definition for what a species is. Actually, most of the time when we mean species in the biological sense of the term, but it is not the only one. And that is because the biological species concept is problematic. And one very clear way to see why it is problematic is that while some species are clearly reproductively isolated from their sister species, remember the hybrids of horses and donkeys are sterile, in many cases reproductive isolation is not complete, meaning that hybrids between some species can occur in the wild which are fertile or at least not completely sterile. And that is very common in nature, for example in birds, with ducks and geese being the sort of the champions of hybridization. But also you find it a lot in insects, for example in butterflies it's been documented, cichlid fish from the great African lakes, or in plants, particularly intensely in what is called syngamians, which are groups of species where the species boundaries are really, really porous to gene flow. So here are some examples to the right of iris flowers from California. And hybrids have been known based on phenotypes only for quite a long time, but the recognition of the prevalence of hybridization has really increased with more and more accessible DNA sequencing technology, which made possible to show traces of past hybridization between species even without clear phenotypic hybrids. And this is done by detecting introgression of genetic material from foreign species into the genome of a given focal species. And thanks to that, we now know that hybridization occurs in many more species than we thought, has been going on for a long time, and has actually happened in our own species. So, for example, we know that about 2% of the genome of Eurasian people comes from introgression from the genome of Neanderthal humans, so a different species of humans, into the genome of Homo sapiens, our own species. We also know that many East Asian and Pacific modern human populations have genes coming from Denisovans, which is yet another species of extinct humans that used to coexist with our own species in Asia. And because hybridization between lineages is so pervasive, it has actually been suggested that a, quote, network of life may be a more useful metaphor than the classical tree of life approach on which most of systematics and phylogenetics, and actually this module of the course, are actually based. Another clear way in which we can see the problem behind the biological species concept is the case of ring species that we have talked about already in the lecture on speciation. When you have a population extending its range to go around a geographical barrier like California salamanders or these greenish warblers around the Tibetan plateau, and the two edges of the range meet on the other side, well, that's the case of ring species then, and by the time those edges of the range meet, both hands have basically accumulated enough genetic differences that they have become reproductively isolated. Well, what about the continuous, uninterrupted sequence of local populations through which genes can still be exchanged all around the geographical barrier? We can say that because the edges are reproductively isolated, that they constitute two species. But can we really say that if the two edges are basically connected by populations that can all interbreed with their direct neighbor? It's just not so clear cut. So that really makes the biological species concept quite wobbly, right? Because clearly many species are not fully reproductively isolated, and so that begs the question, should we revisit which groups we call species so they strictly match the biological species concept? Then if we did that, it's quite possible that we would end up with almost no separate species left because so many species hybridize. But beyond simply affecting the number of recognized species, we have to ask, those groups, like the ducks that hybridize a lot, that are usually considered species but still exchange genes occasionally, are they discrete entities of nature that deserve a special categorization? Well, some people think so and have argued that full reproductive isolation is not the key criterion for species delimitation. And their main argument for that is that despite hybridization being everywhere, we are still able to tell apart the hybridizing species. They remain phenotypically, evolutionarily distinct in some way, even though gene flow is not completely zero between them. And so it has been suggested, for example, that a definition centered more around clusters of genes may be more useful. The genotypic cluster species concept is one such definition, which basically says that despite introgression events, if most of the genes do not end up freely passing the barrier between groups, and therefore groups remain as broadly separate clusters of genes, then those groups qualify as species. In that view, the Neanderthal, the Denisovans, and Homo sapiens, which should technically not be separate species under the biological species concept, because they exchange genes, are nevertheless species under the genotypic cluster species concept, because although they hybridized, most of their genes, of our genes, actually, remained a cohesive cluster. And to be clear, this species concept does not solve the problem of whether ring species are different species, because you can equally argue that there cannot be distinct clusters of genes if there is a genetic continuum between the populations throughout the range of a ring species. Which is to say that even a species concept designed to take hybridization into account still doesn't offer a bulletproof way to overcome the problems brought about by hybridization in the delimitation of species. Another concern to have when coming up with a species definition is that of the systematic validity of the species in terms of taxonomy. What does that mean? Well, species are one of the levels of systematic classification, right? Together with genus, family, order, class, phylum, and all of the things in between, basically. Subfamilies, supraorders in some cases. Of course, it's recognized that these taxonomic ranks are often relatively arbitrary when it comes to how many lineages they contain. For example, the order Carnivora, so dogs, cats, bears, within mammals, has a little less than 300 species going back about 40 million years, while Coleoptera, so the beetles within insects, which is also an order, has 400,000 described species, and the earliest fossils go back almost 300 million years. So clearly, massive discrepancies of what those terms, family, order, class, mean in different parts of the tree of life. But at least these terms have in common that they try to reflect the evolutionary history of the lineages they contain. And here I have to take a little segue through the history of taxonomy and systematics a little bit, because it wasn't always like that. There are many ways of classifying whichever groups we have identified as species. Regardless of the species concept we use, we can classify species based on whether they are useful or pests, or based on whether they swim, run, crawl, or fly, or basically any way we want. There is actually a classification system used in New Guinea by the Rofaifo people, which categorizes species of animals into a few higher groups, but one of them consisting only of eels, cassowaries, large marsupials, and large rodents. Now, of course, natural history has made a point of classifying things together that had similar biological characteristics, right? With mammals lactating and having hair, reptiles having scales and laying eggs on land, fish also laying eggs but with gills, birds flying and having feathers, etc., etc. But we have to remember that these classifications predate the theory of evolution, and even more its acceptance by the general population. And so Carl von Linné, who came up with the binomial nomenclature in the 18th century, actually lived a century before Darwin published The Origin of Species. And so it is only after that that the realization kicked in that if species belonging to a certain taxonomic group looked similar, it was probably because they inherited their characteristics from a common ancestor, and so were probably more related to each other than to species from other groups. So taxonomic categories like family, order, etc. should perhaps reflect this evolutionary history of the things they contain. Now, as you probably already know, a couple of the taxonomic categories in use historically, which were grouping species together based on some superficial characteristics like scales or feathers, turned out not to be very informative when it comes to evolutionary history. For example, from early on, cladistics, and then in more modern times, phylogenetics, figured that reptiles and fish, although they are actually groups that you can make, are not very useful evolutionarily speaking, because they are what we call paraphyletic. They do contain a bunch of species and their common ancestor, but somehow forget about part of the descendants of that common ancestor. For example, some descendants would be excluded from reptiles because they have feathers, and those would be the birds, or hair, and those would be the mammals, which is a bit of an arbitrary cutoff. Actually, the group that contains all reptiles, but also their mammal and bird descendants, is called the amniotes. In contrast, mammals and birds are monophyletic groups. Mammals include all the descendants of the ancestral synapsid lineage that gave rise to mammals, and same for birds, with respects to the descendants of that one line of dinosaurs that led to birds. No downstream branch is excluded. They are informative about evolutionary history. And then we have, just as a note, the infamous so-called polyphyletic groups, which are even less informative since they amalgamate groups with different common ancestors, or put it another way, they do not include the common ancestor to all of the species they contain. So an example of polyphyletic group that was historically in use in taxonomy and systematics is the term pachyderm, which was used to refer to elephants, hippos, and rhinos, so basically big mammals with thick skins, which is actually what pachyderm means, thick skin. Now we know that rhinos are perissodactyls, together with horses, and tapirs, that hippos are cetartiodactyls, together with cows, sheep, goats, deers, antelope, pigs, and whales. And we know that elephants are very, very far on the phylogenetic tree of mammals, since they are not even Laurasiatherians, like perissodactyls and cetartiodactyls, but instead they are Afrotherians, together with manatees and hyraxes. So modern systematics tries to focus on those categories that are monophyletic because that is evolutionarily informative. They try to avoid paraphyletic and polyphyletic groups. And since species is one of the levels of taxonomic nomenclature, it has been proposed that maybe we should be extra careful that species too reflect the evolutionary history of the populations within. And actually, it is not so obvious that the biological species concept takes care of that. You can imagine a situation where, for example, a new biological species arises, diverges, and becomes reproductively isolated from one of the populations inside of a species consisting of multiple populations which can all interbreed. So take, for example, the green species C, as one of the populations within species B, but that becomes a new species because it evolves reproductive isolation. In that example, technically the new species is more related to one of the populations within species B than that population is to any of the other populations within species B. And so technically the parent species, species B, is now paraphyletic. Why? Because it no longer contains all of the descendants of its last common ancestor. It is no longer monophyletic once species C has evolved. According to the so-called phylogenetic species concept, the parent species is no longer a species. The correct phylogenetic species in that case actually is the group of populations that still encapsulates the new reproductively isolated biological species that was just born. So species B and C together, that would be a correct phylogenetic species under the phylogenetic species concept. So this is a problem that has the potential to be very common because reproductive isolation does not need to coincide with monophily. Take sticklebacks, for example, these fish that are widespread across the temperate northern hemisphere and that are very good at colonizing freshwater habitats from their ancestral marine habitat. By doing so, they undergo basically multiple parallel speciation events across the world, forming a new species each time a population colonizes a new river or a new lake from the nearby ocean, and this event repeated as many times as there are rivers to colonize since the recess of the polar ice sheets after the end of the last ice age. So for each newly diverged species of freshwater sticklebacks, you can fairly argue that its closest relative is the neighboring marine population, and that those two are much more closely related than the local marine population is to, for example, another marine population one ocean away, although they are part of the same biological species. These biological species boundaries do not coincide with the phylogenetic species boundaries. The monophily problem is even clearer when we consider a new species emerging not from one population among many, but from certain individuals in the population, as is the case when polyploidization causes instant reproductive isolation and speciation. This is a recurring phenomenon in plants which can tolerate changes in ploidy and remain viable generally better than animals. We have not talked about this mode of speciation too much in the speciation lecture because it does not present much of an evolutionary puzzle for the buildup of reproductive isolation. Because of some malfunction during meiosis or mitosis, proper chromatid segregation fails and an offspring can be born with a different ploidy, so a different number of sets of chromosomes from its parent plants. This usually makes the seedling reproductively incompatible with its parents. Therefore, a new biological species is born. And so wheat and cotton are examples of that, although they are also examples of hybrid speciation, because the ploidy change also involves, in their case, a crossing between species. So they are special cases of polyploid speciation called allopolyploid speciation. They come from crosses between species. Anyway, when reproductive isolation is achieved instantly in a single individual, by definition, that individual is more closely related to its parents than to other individuals in the population. If that individual is classified into another species, then its parent species is de facto no longer monophyletic and no longer a good phylogenetic species. Now, an obvious criticism of the phylogenetic species concept is that the species status of a group of populations depends on whether other populations have gone extinct or have emerged that were or that are related to it. In the previous example, species B was fine being a species until species C appeared. Nothing happened to species B, it's still here and there, and still it is no longer a species according to the phylogenetic species concept at the moment that species C emerges and evolves reproductive isolation, becomes a new biological species, which is a feature that has been deemed not too desirable by some authors in the scientific community. Also, in the same example, species A and species B may not have always been good phylogenetic species. Maybe species A emerged from some population of species B that later became extinct and we don't know it. Does that mean that species E is no longer a good phylogenetic species? It's hard to tell unless we have full information about all the lineages that ever went extinct, which we typically don't have. So we saw that the biological species concept is problematic, the genotypic cluster species concept is problematic, and the phylogenetic species concept is also problematic. Now, how does that work in asexual organisms, which by definition are reproductively isolated from each other at the individual level? Is every individual a new species? Generally, the phylogenetic species concept is easier to apply in asexual organisms than the biological species concept. For example, because we don't have to worry about reproductive isolation so much. But still, at what cutoff should we delimitate lineages into species? Traditionally, in bacteria, it has been common to use a genetic distance criterion above a certain threshold of sequence divergence, DNA sequence divergence, say 3%. Two strains would be considered different species. This, of course, is totally arbitrary and can result in bacterial species that are way more diverse genetically than their eukaryotic counterparts, especially if we consider all the accessory genetic material being passed around through horizontal gene transfer among lineages of bacteria. But that is a story for another lecture in another module. Actually, to make clear that the threshold-based bacterial species concept is arbitrary and just aimed at being operational, rather than pretending to distinguish between real, well-separated entities of nature, microbiologists talk about operational taxonomic units, OTUs, instead of species proper when using these species delimitation methods. And to really take the point home that indeed species delimitation may be more of a headache in asexual species than sexual species, it was actually shown in rotifers, these microscopic animals, some of which, the bdelloid rotifers, are asexual, Others are like the monogonont rotifers are sexual. It was shown that indeed populations in the asexual clades form less discrete, less clearly delineated genetic clusters than in the sexual clades because of the absence of removal of intermediate hybrid genotypes during the process of reproductive isolation. And I refer you to the speciation lecture to understand what that means. There are basically more intermediate genotypes in asexual species than in sexual species, which has even led some authors to suggest that species may simply not be as much of a thing in asexual organisms at all. Okay, so far we've seen a bunch of different species concepts, all of them requiring knowing about the reproductive compatibility or about the genetics of the potential species. But what if you don't have that? And indeed, we don't have strict measures of reproductive compatibility or genetic data for most species on the planet. Well, that's where we talk about morphospecies, which are identified based on phenotypic characteristics. Most species ever identified are actually only ever identified on the basis of their phenotype. Think, for example, of all the 400,000 species of described beetles that I have mentioned earlier. Most of them are morphospecies, and for good reasons, since species do indeed often differ in visible traits. Before DNA technology, for example, bacterial species were identified on the basis of the shape of the colonies that they would form. Of course, the fact that most species are identified as morphospecies does not mean that they are not also biological species or phylogenetic species. We just haven't checked. Many times morphospecies turn out to be also fairly or completely reproductively isolated, but other times no. And this is exactly what helped identify the problems with the species concept. Many established morphospecies were identified as exchanging genes, for example, and yet remained perfectly identifiable based on phenotypic traits. Thanks to genetics, however, it has become possible to find so-called cryptic species, so species that were thought to be the same based on phenotypes, but are actually fairly differentiated genetically, suggesting some high levels of reproductive isolation. This happened recently, for example, in anaconda, where a new species was discovered, and the headlines of the regular news were a bit misleading, saying that a new species of giant snake was discovered in the Amazon rainforest. What actually happened is that those snakes were already known. They just figured that you had different genetic clusters that push people to describe a new species of anaconda. The giraffes are also another example of that. Started as one species with 9 subspecies, now split into 4 recognized species across the African continent. In general, the discovery of cryptic species leads to more subdivision of the existing recognized species, causing a phenomenon that's called taxonomic inflation, so an increase in the number of recognized species through time, not because we discover new organisms and new forms, just because we decide to split them more. And this has consequences for conservation biology. You can imagine that if all of a sudden what we thought was one species of giraffes are actually 4 species, or in other cases like subdivided in even more species, then each of the new species that don't really exchange genes with the other taxa that have just been elevated to the species rank, well, then each new species might be much smaller than previously thought, much more depauperate in terms of genetic diversity, and maybe more at risk of extinction. in the face of some environmental disturbance. Now, that was a case where multiple biological species sort of hide within fewer morphospecies, but it can also be the other way around. And there are cases where there are more morphospecies than there are biological species. For example, in fossils, and fossil species are by necessity only described as morphospecies. Specimens classified into different species because they look different may actually belong to the same biological species, just maybe be of different ages or different sexes. For example, there used to be 16 species of Triceratops since its first description in 1889, and now there are only 2 convincing recognized species left. But of course, because we only have access to morphology and no other phenotype with fossils, it could be that there too we are missing cryptic species that would have the same skeleton but have, for example, very different color or ecological niches, and definitely some reproductive isolation. We just wouldn't know it. Tigers and lions, for instance, have very similar skeletons and similar sizes, and you really need to look closely at small details in the skull to tell them apart. And if a paleontologist from the future were to find some slightly incomplete skeletons, they may just not be able to tell the 2 species apart. And so the problem of how many biological species there are behind morphospecies can really go both ways. So yeah, that was an overview of the various species concepts. I want you to retain 2 key messages from this lecture. First, what is a species is a complicated question. Some have argued about what the best species concept might be, and here you saw a few in this video, but there are actually close to 100 different proposed species concept, and so there are many. I'm throwing in there the ecological species concept as well, which I have not mentioned too much, but it's a similar one to the morphospecies concept. Just identifying species as groups of populations that are occupying distinct ecological niches. And I'm also adding the evolutionary species concept, which basically treats species as entities that evolve independently from each other. It is not very commonly used because it doesn't really make explicit predictions about the criteria used to distinguish species, unlike the biological species concept or the genotypic cluster species concept. What is clear is that although some concepts are way more popular than others, for example, the biological species concepts or the genotypic cluster species concepts are still mostly implied when we use the word species. Despite that, no concept is bulletproof and without caveats or without limits to its domain of validity. So the debate remains open, but has relatively settled down, with people realizing the limits of our attempts at discretizing something that in a sense might be more continuous at times, and that maybe different definitions may be more relevant in different situations. Some even think that the debate is more about semantics at this point than about actual biology. The second message I want you to take home is that the fact that the species concept is a mess does not mean that speciation cannot be studied. Of course, we know that even when speciation results in completely reproductively isolated species, at some point in the past, that was not the case. And at some point in the past, it wasn't clear whether we had 1 or 2 species. So, and it's equally unclear at what time point along the speciation continuum, at what time point we can say that we have two species. Regardless of that, speciation as a continuous process that generates diversity through divergent selection and in some cases promotes the rise of reproductive isolation happens anyway and can totally be studied even without setting a clear boundary from which we say we have two different species. And so perhaps paradoxically, the study of the formation of species does not necessarily always rely on adopting one specific species concept. So yeah, I'll leave you with that on this video on species concepts, and thanks for watching.